study of the production of the like penicillin, citric acid, vitamin B12, glutamic acid, and this thing called Griseofulvin. So I think I have started the discussion with the penicillin. So for penicillin, we already have learned in detail in our first unit, where in detail we have learned what is penicillin is, how penicillin was accidentally discovered by the great scientist called Alexander Fleming, and penicillin was the first antibiotic in the history of our mankind. And all the penicillin, you will be getting such kind of beta-lactam ring through one video. I have shown you how penicillin is actually stopping the synthesis of the bacterial cell wall. बैक्टीरिया की सेल वॉल जो है उसका सिंथेसिस कैसे वो बंद कर देता है वो हमने देखी. Then the question was that how you will be able to produce the penicillin. So these are the various properties. I think that part also I have mentioned. And for the biosynthesis, you can follow this particular flow that is a workflow. You can start with the L-alpha amino adipic acid, and through various steps you can ultimately getting penicillin G. But there are some matters, some questions that all the penicillins are actually not get dissolved into the water. So that's why earlier time, at the beginning when Alexander Fleming have discovered the penicillin, that time penicillin was actually giving to the patients through in vitro, through intramuscular, okay, intravenous, because penicillin actually not dissolved into the water. If something is not dissolved into the water, some drug, you cannot take it orally directly, because it has to be passed through your GIT, gastrointestinal tract. If it is dissolved into the water, then it will very easily get absorbed by your intestine. So that's why later on we have seen there is another version of penicillin V. But as of now, you can produce this process. Then how penicillin is working, already we have seen that thing in our previous lecture. Then there are various types of penicillin. One is the penicillin G and penicillin VK, methicillin, neficillin, ampicillin, and amoxicillin. Okay, these are various types of the penicillin. I think ऋषभ ये part भी मैंने discuss किया था in previous lecture.
Yes, sir.
Okay, this part upstream and downstream.
बता दो.
Okay. So for the production of your penicillin, you can go through this process. So in industrial production, penicillin are actually broadly classified into two different ways. One is the upstream process, one is the downstream process. Now what is upstream process? Upstream process actually it is encompasses the any technology that leads to the synthesis of the product. Leads to the synthesis of the product. I think in medicinal chemistry you are learning from your teachers that how you can synthesize a product in your lab. So the upstream actually including the exploration, development, and production. Exploration. If you have a beta-lactam ring, you get to explore how you can change this the moiety are actually attached with the beta-lactam ring. So exploration. You can develop and into the production. Now what is downstream? Downstream is a process of the extraction and purification of the biological product from fermentation. Refer to the downstream. For example, you have a fermenter. You have a fermenter. And here you have the pedal and all the propellers, etc. So here, the very natural process, you have produced some this thing called product. You have produced some fermented product. Penicillin you have prepared. Now the penicillin whatever you have prepared here, it is not completely pure, or it is not completely at the correct form, so that you can take to this component into as a drug to the patients. You can give this substance as a drug to the patients. It is not so much accurate. So first you will do filtration and you will do other kinds of activity, but after that you can further modify that thing. After that, if you can modify that thing, okay, that process is actually called as a downstream. And in upstream process, if you add some technology, where in this process when it is actually just revolving, and when your bacteria and other substances are actually here, at that process if you make some changes, then it will be called as a upstream process. So yeah, am I audible to you, students? Rishabh?
Yes, sir.
A sudden disconnection of internet. Okay. Anyway, so I hope you can remember that for the culture method for fungus, we have actually discussed earlier about the Penicillium notatum. Penicillium notatum, which is one of the fungus. So Penicillium notatum can be used as a culture. So you can that is a do in two methods, a surface culture method or submerged method. ओके सबमर्स्ड याद है सबको सबमर्स्ड क्या होते हैं हम लोगों ने पहले भी देखा था अबाउट द सबमर्स्ड एंड वी आल्सो हैव सीन द सॉलिड स्टेट फर्मेंटर okay or sometimes we also call it as a surface culture method. So in surface culture method fungus is actually cultured on the surface of the liquid medium without any agitation. After an appropriate incubation period penicillin actually extracted from the medium and this is a old method. Okay, this is a very old method. What is submerged method? What we are doing simple, whatever I was actually showing you at that time. If it is a fermenter everything will be submerged, one agitation will be there, your fungus and your medium will be in submerged condition. So submerged culture method the fungus actually grow in a liquid medium which vigorously agitated and aerated. After an appropriate incubation period the penicillin is separated from the medium and today penicillin is actually produced by this submerged method. Okay, so that's a great thing. In my previous lecture several times I have told you why that is a solid state fermenter actually much more preferred than a submerged. But you might be wondering to see here that in terms of penicillin we're actually that is giving preference to submerged culture method. Yes, that's true. You have to understand that whatever the procedure we may follow, solid state or submerged, whatever procedure we follow, we shall follow based on our requirement. There might be some reasons why scientists are actually preferring the submerged method for the penicillin. Okay, so here is the entire flow chart. So you have to prepare the medium, then fermentation will be happen. After fermentation, you will be collecting the product. Then you have to centrifuge that thing and then you will do filtration, then solvent extraction will be there. After filtration whatever the remaining things will be present from that you will be extracting the solvent and then you will precipitate and after that you will be getting some crystal product, some crystal powder format, powder product, and that powder you can take convert into that crystal into tablet or if you can also convert into the capsule. Okay, SULE. Earlier time after filtration directly people used to after filtration people have to go for the solvent extraction and then people used to take the solvent and that solvent used to give as a intravenous IV, intravenous route as an injection earlier time. But nowadays it is so much difficult ना injection लेना कौन पसंद करता है अभी जो है right now the vaccine COVID vaccine is actually giving in the form of injection. So all of the people are not happy to take injection. So it's a problem, patient compliance. So that's why in present scenario we're actually taking two more steps, this and this, and we are bringing into the capsule and tablet form. So what are the media formulation for the penicillin? So for that you need to the pH up to 6.5, temperature up to 20 to 24 degrees centigrade, oxygen you need, nitrogen you also need, glucose also need, then ethanol, then phenyl acetic acid, and this thing called probenecid. Now you might be wondering that sir why pH is actually acidic, slightly acidic, na 6.5. So you have to understand that whatever the product actually will be generating, so based on that product and based on the correct environment of the fungus, we need to create an environment. For that you have to provide such kind of things. Oxygen, nitrogen, and temperature should be up to this and pH up to this. If you vary this thing, all of you have seen in my previous slides I have mentioned that variation of the factors what will be happening. So it will be not able to produce sufficient amount of the penicillin. Penicillin production will be get degraded or it will be get hampered. So that's why you have to ensure that thing. Remaining things like glucose, ethanol, phenyl acetic acid, and probenecid you have to provide. So as a component, so that they can take those substance and they can do the fermentation. Including lactose and calcium carbonate you have to also provide, and yes, including anti-foaming agent, vegetable oil. I hope you can remember I have also shared one of my experience, a very bad experience with foaming है ना. जब मैंने पहले बताया था तुम लोगों को, when I was in industry, I have faced a very difficult time one day in industry. I was producing some substance that I cannot reveal in the lecture. So the foams was actually forming in a very rapid scale. I informed to my immediate senior manager that forms are actually presenting forming. So it was a very big problem. At that day, we did not have the anti-foaming agents, but somehow we actually completed the work. So that's why you have to ensure that there should be no any foam should be appear. And after that, you also have to do heat sterilization at the high temperature. For example, in this machine, the things actually happening. See, why you actually doing heat sterilization? See, you're actually using the bacteria, the fungus, Penicillin notatum. If any bacteria or if any virus or if any, there are lots of bacteria are present which are actually harmful to the fungus, which are actually killing to the fungus. There are some viruses or other microbes are also. So you have to kill all those things so that your fungus can grow very rapidly. Then after that, fermentation will be start. This is the various parameters. All of you are already have seen the temperature and the pH, including the pressure, and the batch. You can go with the batch fed mode or high amount of glucose actually you need for the production of the penicillin. So here you can see the various fermenter. Very beautiful pictures are here. You can see. So यहाँ पे जो है सारा activity in this big fermenter will be happen. Okay, the remaining thing what we are actually observing, these are the associated components. They are either providing oxygen, they are actually giving nitrogen, carbon dioxide actually being removed, and other whatever the activities, real time monitoring, they are actually taking a big part. So during discussing with you how you can produce a large scale fermenter, I have told you the importance of the impeller and asperging. So seed culture, so first done into the lab by adding the penicillin spore in the liquid medium, and after that it should be inoculated into the fermenter. Seed culture means I like to tell you that for example penicillium notatum you have to culture that thing, and after that you have to place those things here. Okay, this is called actually seed culture. Then after completion of your entire work, you have to filter. After completion of the entire work, you have to filter. These are the rotary vacuum filter. There will be rotation. It will be actually rotated so that the filtration can happen in a very systematic जो है manner. यहाँ पे तुम लोगों को समझना होगा जो यहाँ पे प्रोडक्ट बनेगा ना आफ्टर एंटायर द एक्टिविटी पेनिसिलिन के जो प्रोडक्ट बनेंगे, दिस प्रोडक्ट विल बी सो मच दैट मीन्स क्या कहूँ मैं, गाढ़ा होगा काफ़ी. इट विल बी सो डेंस. डेंस दैट्स वाई इन सिंपल फिल्ट्रेशन जो तुम लोग घर में जो है यूज़ करते हो ना फॉर द फिल्ट्रेशन, लाइक क्या कहते हैं इसको, छकनी कहते हैं बहुत टाइम, है ना. इट विल नॉट एबल टू यू कैनॉट यूज दिस काइंड्स ऑफ होम like that is a filtration equipments. You have to go with the rotary vacuum filter. Then you can also add some solvents, and after that you have to do centrifugation so that if any cell components or other wastage are actually remaining, it can be eliminated, and then you have to extract the actual penicillin. So with penicillin you have to add some acidic solution and with the various buffer to maintain the pH of the particular substance, that is penicillin. So this is the entire process. You can see in this picture also. This is the basket centrifugation machine, and then you can go for the fluidized bed dryer. Okay, to remove the moisture present into the penicillium salt. Basically, we are actually getting this penicillin into a that is a solid form. Okay, so to remove the moisture content, you can go for the fluidized bed dryer. Hot gas are actually pumped from the base of the chamber, and powder salts is also contained into the vacuum chamber, and result should be come into the dried from the penicillin. Dried from माने powder आप form होके आएगा. And after that you can store into the various container. Either you can, whatever I have told you in the previous slides, either you can pack into the liquid or into a solid penicillin pills. Right now the process is not so much that is useful because a lots of bacteria are actually getting that is resistant with the penicillin. We are also working on this kinds of problem. We also have some project. One of the project that I am right now working on, bubonic plague. I hope many of you might have remember or might have recently got to know there is one dangerous disease called bubonic plague. So it was the first that is a pandemic in the world. So we are also working on this disease. We already completed 80% of the work, and we are now at the stage of the manuscript submission. I hope very soon you will be able to see one of my research paper publication on the antimicrobial drug resistance called bubonic plague. This is the entire process. You can do all those things, filtration and another thing, and lastly you'll be getting these things. So advantage, already you are aware that it has have a excellent tissue penetration, bactericidal against the various strains, and relatively non-toxic. Penicillin is actually non-toxic. You know why? Because penicillin कहाँ पे काम करता है? Penicillin is actually stopping the synthesis of the bacterial cell wall. Now, all of you might have seen that video. There was some enzyme called peptidase. So जिस enzyme में वो काम कर रहा है, that enzyme is actually not available in human. हमारी human body में जो है वो particular enzyme present ही नहीं है. ठीक है? That's why this enzyme or this particular substance is present only in bacteria. So even if bacteria is entering in your body, and if you give penicillin, so your drug will be only bind with this particular that is enzyme. It will not bind with any of the human protein. That's why it will not show any kind of side effects. All of you are saying that sir, many of the drugs are showing side effects. Many of the drugs are showing side effects, including that is a famous Indian Ayurvedic that is a brand ambassador like Swami Ramdev. Swami Ramdev usually he says that in allopathy there is a big problem of the called side effect or drug interaction. Actually, the problem is that today, whatever the drugs we have, now almost 20,000 of the small molecules, whatever we have. So those molecules, when it was discovered, they have not done any proper study of the off-target binding. Off-target binding means you are trying to block a particular virus protein. You are trying to block a particular viral protein so that the virus cannot replicate. But during that time, you have not done any kind of study that whether that drug is also binding with human protein or not. For example, penicillin, it is only binding with the bacteria's cell wall. It is not binding with any of the human protein. Okay, that's why it is actually working. It is not showing any kind of proper such kind of problem, side effects or other things. So in future, when you will be also doing drug discovery and other kind of activity, you should take care of this matter so that you can get the better drug which is not showing any side effects. And yes, it is efficacious in the treatment of the infection and inexpensive in comparison with the other antibiotic, and newer penicillins are actually resistant to the stomach acid such as penicillin V. A broader spectrum such as ampicillin and amoxicillin. I hope amoxicillin, the name is very much common to you all, because many of time you might have taken also amoxicillin. There are some disadvantages also of the penicillin. One of the disadvantages is the acid liability. So most of the drugs actually destroyed by the gastric acid. It's a big problem. So can you tell me what are the actually acids are present in our gastric acid or GIT? Anybody? ऋषभ, can you tell me? Pinky?
No, sir.
आई तुम लोग पता नहीं ये चीज? All of you are aware that thing. हमारे पेट में कौन से एसिड रहते हैं? Our stomach? बताओ. Can you hear me, Pinky? Stomach में कौन से एसिड रहते हैं?
Sir, bile acid.
हाँ यस, बाइल एसिड. उसका कुछ और बताओगे और डिटेल्स में? कहाँ? हाँ यस, और कुछ बताओगे?
HCl होता है सर.
हाँ बताओ क्या होते हैं?
HCl होता है सर, HCl.
Yes, absolutely, Ravi. Ravi is very wonderful. He always give the correct answer. Since last many of the days, I am observing. Very good, Ravi. HCl. सभी की जो है in our that is a GIT, we have HCl, hydrochloric acid. So you shouldn't forget that thing. These are the very basics. ठीक है? फार्मेसी एंड आवर बायोटेक्नोलॉजी जानते हो, इतना अमेजिंग सब्जेक्ट है, इतना अमेजिंग. अगर तुम एक बार बेसिक्स को एक बार तुम दोबारा रिवाइज करके पढ़ोगे ना, ये सब्जेक्ट बहुत ही अमेजिंग लगेगा. and you definitely become a scientist, definitely. जानते हो प्रॉब्लम क्या है? प्रॉब्लम ये है हम बच्चों को बस एग्जाम पास करने के लिए पढ़ाया जाता है। सो एग्जाम पास करने के बाद सबके पास होता है डिग्री। अब डिग्री देख के जो है कोई नौकरी देता नहीं है। इट्स ए ट्रू फैक्ट, ट्रू रियलिटी ऑफ आवर कंट्री, नॉट इन कंट्री, ऑल ओवर द वर्ल्ड। सबको चाहिए होता है नॉलेज। Right now, the new diseases are actually emerging, and new kinds of disorders are actually coming. That's why a big activity is actually going on the research. So if you want to go in research in future, if you want to become a scientist, you have to go through with your previous knowledge the basics. ठीक है? तो एनाटॉमी फिजियोलॉजी तुम लोग पढ़े थे, बट तुम लोग भूल गए, इंक्लूडिंग, देखो मैं इसलिए भी कह रहा हूँ, देखो तुम लोग अब अगले कुछ टाइम बाद ऑल ऑफ यू आर गोइंग टू गिव सम वेरी इंपॉर्टेंट एग्जाम लाइक जीपेट, ठीक है ना? जीपेट एग्जाम तुम लोग दोगे। सो इन जीपेट एग्जाम यू नीड द बेसिक्स नॉलेज। तो जैसे अभी मैंने पूछा, तो जो है तुम लोग थोड़ा जो है यू आर नॉट एबल टू गिव मी द फ्लुएंट रिप्लाई कि सर इससे, बिकॉज बहुत पहले पढ़े थे, अभी याद नहीं आ रहे हैं ना, ऐसा रीजन रहा, आई कैन अंडरस्टैंड। सो यू शुड रिवाइज एवरीथिंग, एंड आई विल सजेस्ट राइट नाउ इट इज़ द मंथ ऑफ जुलाई। सो अगले साल आई थिंक जब तुम लोग अगले 1 साल या 1.5 साल में तुम लोग जीपेट एग्जाम दोगे, सो यू शुड प्रिपेयर साइमल्टेनियसली विथ योर सेमेस्टर एग्जाम्स। जो सेमेस्टर की पढ़ाई कर रहे हो उसके साथ साथ जो है जीपेट की पढ़ाई भी तुम लोग करते रहो। इट्स वेरी इंपॉर्टेंट, एंड यू शुड क्लियर एटलीस्ट योर जीपेट। उतना टफ नहीं है, हवा बना के रखे हुए लोग, ठीक है? कुछ बेसिक्स बुक्स है, वन डे यू मे आस्क मी, आई विल टेल यू हाउ यू कैन दैट इज प्रिपेयर। इट्स वेरी सिंपल, यू नीड सम बुक एज ए synopsis, you need one book for synopsis like पीयूष की एक बुक है, तुम लिख सकते हो। और एक बुक तुम्हें चाहिए वो एमसीक्यू के लिए। एमसीक्यू के लिए दुनिया भर की बुक है, ईमानदार और काफ़ी सारे बुक भी है, IMANGR, आलियर सर, काफ़ी सारे बुक्स है। दो बुक्स जो है तुम्हें चाहिए होगा। हर दिन जो है, हर दिन तुम एटलीस्ट 40 पेजेस जो है पढ़ो। अब जब मैं कहता हूँ 40 पेजेस, तुम लोग कहोगे कि सर इतना 40 पेजेस हम लोग इतना तो नहीं पढ़ते। देखो हैबिट बनाओ, विथ हैबिट। जानते हो मैं क्या बताऊँ तुम लोगों को, बिल गेट्स है ना, बिल गेट्स इतने जो रिच आदमी बने वो कैसे बने? बिकॉज बिल गेट्स रीड्स 200 पेजेस पर डे, और वो काफ़ी लेट से पढ़ाई शुरू किए। तो अगर तुम्हारी एज में से बिल गेट्स अगर पढ़ते होते तो बहुत आगे निकल गए होते। तुम्हारे पास टाइम है, तुम्हारे पास मौका है, और पढ़ना एक हैबिट है, इट्स जस्ट हैबिट, ठीक है? एक बार तुम 21 दिन 21 डेज, आई ऑलरेडी टोल्ड यू वन डे देयर इज अ रूल ऑफ 21 डेज। कोई भी काम 21 दिन तक करो तो तुम्हारी हैबिट बन जाएगी ये चीज़। अर्लियर जो है, मैं अपना भी एग्जाम्पल दूँ, पहले जो है मुझे सुबह उठने में बहुत दिक्कत होता था, आई वॉज ए लेट राइजर। जब से मेरी मम्मी यहाँ पे नोएडा में आ गई है, तो एवरी डे शी एक्चुअली फोर्स मी टू वेक अप एंड गो फॉर दी दैट्स मॉर्निंग वॉक। तो अब मेरा आदत हो गया है, अब हर रोज़ 5:30 बजे मैं जाग जाता हूँ। सो हैबिट, ओनली 21 डेज यू हैव टू फॉलो ए स्ट्रिक्ट रूल ऑफ ओनली 21 डेज। 22 दिन से जो है 22nd डेज से तुम्हारा हैबिट बन जाएगा। सो दैट्स वाई आई विल सजेस्ट दैट यू मे just revise your old the concepts, the basic concepts. You can take some books like synopsis, जीपेट synopsis by पीयूष। उसमें पुराना जो है जो तुम लोग पढ़े हो, जो फर्स्ट सेमेस्टर सेकंड सेमेस्टर सारा कुछ वहाँ पे होगा, जस्ट एक बार रिवाइज करो, और उससे जो है तुम्हारी बेसिक्स उतना स्ट्रांग होगा। सिर्फ जीपेट ही नहीं, कोई भी एग्जाम हो तुम लोग बहुत अच्छे से क्लियर कर सकते हो। ओके, so including some lack of the activity against the gram negative organisms, short duration infection, and many patient experience the GI upset, meaning gastrointestinal upset, and painful if given into intramuscularly. That's why nowadays it is giving orally. These are some disadvantages. So yeah, these are the various microorganisms which are actually used for the production of the penicillin. And right now, let me take to you another very important substance called citric acid. So, can you tell me what is the natural source of citric acid? Again, a question from basics. Many of you will be not happy with me. Sir, आज आप ज़्यादा क्वेश्चन पूछ रहे हो हमसे. Can you tell me what is the natural source of citric acid? कहाँ से naturally हमें मिलेंगे citric acid?
Citrus fruits.
Yeah, very good. Any example?
Nimbu.
Or?
Orange.
Where is Pinky today? अच्छा, Pinky तो आंसर दे रहा है. Okay, okay. Where is Rishabh today? Rishabh is...
Tomato.
Yeah.
सर यहीं पे हूँ मैं आज।
अच्छा, ठीक है, very good, very good. So citric acid production. So citric acid is very important for us for various purposes. Now, if you want the citric acid in a very large scale, so you have to take the knowledge of the biotechnology. So what Pinky was actually saying that naturally it is already available in the various citrus food. So from there we can get the citric acid. But if we need heavy in a very large scale, बहुत ज़्यादा अगर हमें चाहिए, तब तो जो है in that scenario we cannot rely on the natural sources, है ना? That's why we are actually taking the help of the fermentation. So fermentation is the most economical and widely used technology for the synthesis of the citric acid production. So industrial citric acid production with three ways. One is the surface, one is submerged, and the solid state fermentation. So surface fermentation. All of you are aware what is surface fermentation. So in that case, we are actually using the Aspergillus niger, and that may be done on rice bran, as in case of in Japan, or in liquid solution in flat aluminium or stainless steel pans. ओके देखो एस्परजिलस नाइजर को जो है यूज किया गया as a substance, as a that is a component which actually get fermented by the as a microbe which can ferment these things, rice grain and other things in Japan. So special stains are also you can use. You can also modify by using the RDNA other technology of Aspergillus niger, and it can produce citric acid despite the high content of trace metal in the rice bran actually used. Okay, so you can either go for the surface or you can go in the submerged method. All of you are aware. Again, I do not want to that is explain because it will be so boring, है ना? काफी बार जो है हम लोग बात कर चुके हैं इसमें. So कुछ नहीं, बहुत ही simple है. Submerged में हम सारा कुछ जो है submerged रहेगा, and it will be submerged I think for 5 to 14 days. After this temperature, 27 to 33 degree centigrade. So it will be produced in the that is a fermentation tank if it is actually purified. Or you can go with the very simple technique, whatever I have actually told you in my previous lecture, called solid state fermentation. It is a simple method for the citric acid production. So the citric acid production actually reached a maximum up to 88 gram per kg dry matter when fermentation actually carried out with the cassava having initial moisture up to 62% at 26 degree centigrade for 120 hours. माने कितना दिन? 24 कितना दिन होगा? almost 5 days, है ना? So solid state fermenter also known as the koji process because it was actually used in the Japan. So the name koji, it is actually Japanese word. So you can follow any of this process, and after that you have to separate by your filtration. Okay, the process सारा जो है सभी में same है. You are taking one microbe and you are taking some component or some media. You are mixing those things either solid state or the submerged, and then you are actually getting some product. You are going to filter that thing, and after filtration, basically filtration separation एक ही चीज है. After filtration, you are getting this final product. If you are interested, you can do some further activities. So this is the entire process of the separation, and then you will be going for the purification. So this is the entire chart of the citric acid production. So you can take the Aspergillus niger, and that thing should be grow into that thing, and you can take some substrate, and then you will be mix the water into a different concentration, or वहाँ पे जो filtration and shellation will happen, and then inoculum. And then after that the production will start, and then filtration, and then cell biomass will be removed, and here you are getting the citric acid. After that you can go for the purification. Okay, this is a simple process. So what are the factors? Again the same things मैंने जो पहले बताया था, the pH, temperature, nitrogen source, aeration, trace elements, these things will be are the factors. So whatever the things actually I am showing you, you can let me just a second. Yeah, so whatever the substances I am showing here, you can produce the same thing, same process. There will be some factor, there will be some process, some microorganism, some particular that is a media. You have to take both the things into your fermentation that is a chamber. You can do either solid state submerged method. After that you are doing filtration, and after that you are getting your product, and then you can go for the purification, or you can do further process like converting into a solid as a powder form, or you can go to the liquid as a liquid form. Okay, so yeah, so that thing I was actually trying to show you. So in industry, same process. So you can use the citric acid in a various way. So for food and drink as a preservative or a flavoring agent, also as an emulsifying agent in the ice cream. How many of you are aware that thing? Citric acid can be also used as an emulsifying agent into the ice cream. At the household cleaner also, kitchen and bathroom that is spares also. So many of time you have seen that your mom actually removing some stain from your clothes or somewhere other else by using the this thing called lemon or the citric acid. You know, my mother is actually using a lot of citric acid in various that is work. Okay, you can also use as a cosmetics. Yeah, that thing I was actually trying to remember. My mom even made the homemade shampoo, okay, by using the simple thing like honey and your this thing called citric acid means nimbu, another thing. It is homemade shampoo. So I was shocked. Is it possible?
सर
Then
कैसे बना था?
हाँ हाँ बताओ ऋषभ.
ये बना था शैम्पू?
हाँ 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 yeah. So actually, in YouTube there are lots of videos. So I also have checked, and then I compared, and I found wow, this is so amazing. So you can use these things also as a shampoo. Okay, then body wash also, and also as a wash cleaner, nail polish, and I think as a hand soap or other cosmetic product. So that's the thing. That's it for citric acid. Now let me also tell you a few things about the vitamins. So vitamins, all of you are aware. After COVID, many of you might be taking vitamin C, vitamin D a lot, you know, because including vitamin B12, because in various that is a research paper, the scientists have mentioned that nutraceuticals can play a very big role in boosting your immunity. So here I have got a very interesting picture of the various vitamins and their natural that is sources. Okay, vitamin A, vitamin B1, B2, B3, B5, 6, and 9, 12, C, D, E, K, and BP. So, in all those vitamins, our interest is actually vitamin B12. Our interest is actually vitamin B12. This is also called as a cobalamin. Okay, so let me just take you some very basic things. So you need beta vitamins for your body growth. Okay, so they are actually they have a very great value into the growth of the metabolism into the living cell. And vitamins you can obtain from the food, whatever I am showing you here, and human can also produce some vitamins as a precursor. They consume, and while certain microorganisms also produce vitamins. So can you tell me a name of a vitamin which actually that is got synthesized in our skin from the sun rays? हमारे स्किन में जो है एक विटामिन बी सिंथेसिस होती है.
D.
D3.
या वेरी गुड विटामिन डी. यू नो भारत में जो है नॉट ओनली इन भारत, आई एम ऑलवेज गिव द एग्जांपल ऑफ इंडिया, इन अफ्रीकन कंट्रीज कोविड जो है ना, कोरोना वायरस में जो है इतना तबाही जो है हुआ. अमेरिका में सबसे ज़्यादा लोगों की डेथ हुई है, बट अभी एज ऑफ नाउ शायद इंडिया का नंबर जो भी हो, वट एवर द सिनेरियो, अमेरिका, इंडिया, ईरान, चाइना, रशिया एंड यूरोपियन कंट्रीज, maximum of the people actually died because of COVID-19. Have you wondered that why very less people are actually died in your African countries? अफ्रीका जो है ना, अफ्रीका में तो बहुत खराब हाल है पहले से. The health infrastructure is very poor, but the people in Africa they are actually not so much very easily got infected by COVID. बहुत कम लोग की डेथ हुए. The big reason is the vitamin D. Vitamin D actually helping to boosting the immunity. I will tell you more in when I will teach you the unit 3. Very interesting things you'll be getting there. So in these African countries, they are getting the sun rays directly. वहाँ पे जो है ना सूरज जो है एकदम सीधा वहाँ पे जो है they are getting. These countries are actually situated in the Mediterranean, if I am not wrong, and the equator के आसपास जो है वो countries जो है situated है. So they are getting the sun rays at the high amount. what the people of the cold countries like China and the America, Canada and other countries are not getting. ठीक है, especially the white people. जो white and black है ना, जो ये problem हुआ, what people skin, the big thing actually melanin. When as lots of sun rays you are getting, to protect your skin, melanin will be actually get synthesized and it will be deposited in your skins, so that your skin can be protected because of the presence of the melanin. So African people जो है उनकी skin में melanin ज़्यादा है. We Indians हमारे जो है, we are at the middle. We are not in at the very high temperature based country, and we are not very low temperature. So we are at the middle. That's why we are brown, and we are the very amazing people in all over the world. दुनिया में कुछ भी हो जाए, इंडिया को कुछ नहीं होगा. पहले भी जो है इबोला, ये सारा बहुत कुछ हुआ है. You have seen we are actually very much protected from all the various problems. फर्स्ट वर्ल्ड वॉर हुआ, सेकेंड वर्ल्ड वॉर हुआ, इंडिया में कुछ नहीं हुआ. So that's why as a citizen of India you should be very much proud. And most important thing, देखो सच्चाई तो है, तो मानना पड़ेगा ये चीज़. Despite the इंडिया ने कैसे काबू पाया, जानते हो, ये तुम लोगों को जो है ना जानने की जरूरत है. All of you are supposed to know these things. चाइना में very क्या बताऊँ मैं, they have imposed very severe lockdown. चाइना में जो है बहुत ही सीवियर लॉकडाउन जो है ना लगवाया. इंडिया में जो है उतना सीवियर लॉकडाउन नहीं था, जो इंक्लूडिंग फर्स्ट वेव, इंक्लूडिंग सेकेंड वेव. सेकेंड वेव के टाइम में एट द बिगिनिंग देयर वाज सम शॉर्टेज, बट द वे वी एक्चुअली कम आउट फ्रॉम द सेकेंड वेव, देखो ना अभी खत्म हो गया सेकेंड वेव ऑलमोस्ट. The way actually come out, it was very successful story of biotechnology in the history of mankind. भारत में जो है हेल्थ इंफ्रास्ट्रक्चर कंपेयर टू अदर कंट्रीज वी हैव नथिंग. ठीक है, वी हैव नथिंग, एंड आवर गवर्नमेंट इज नॉट लाइक ए गवर्नमेंट ऑफ चाइना. गवर्नमेंट जो है कुछ भी करे, हम लोग गवर्नमेंट के रूल्स को मानते नहीं है, बट स्टिल वी आर सक्सेसफुल. So it's a very great achievement, and you have to understand that in our old knowledge, that means Ayurveda. So we are still practicing Ayurveda. In terms of, many of you might be taking Chyawanprash at your home. Many of you might be doing a lots of that is natural remedies like haldi and other things. So haldi में क्या है? haldi में turmeric. So that things are actually helping us for boosting our immunity. So, there are lots of things I can say. It will take so much long time, and time will not actually suggest me. So one day I will tell you why we are actually so much successful. Anyway, so for any kind of vitamins, for the preparation of the any kinds of vitamin, you can go through the fermentation process. So the process is very similar. Whatever I am actually telling in the previous two cases. So you have to prepare the medium. You have to prepare the that is a medium. So and then you have to inoculate with your microorganism. Then you have to go for the production medium, then sterilize, and then the main thing called fermentation. This is the main thing, and then centrifugation. After that it will be produced. Then biomass you have to remove. Then solvents जो भी बनेगा, you have to that is a you have to dissolve that particular solvents, and then drying. Then you have to dry the substance. This is a simple process. Here I have written penicillin. So in that case you might be use the vitamins. So vitamin B12, also known as the cobalamin, is a water soluble vitamins, has a very big role in normal functioning of the brain and the nervous system, including formation of the red blood cell. So it is actually synthesized with the microorganism, the same process whatever I actually telling you. If anybody having some deficiency, he will be facing some kind of anemia. Okay, so this is the step. See, everywhere you will be observing same thing in a different that is in a very different presentation. Same thing. See, fermentation of the medium, sterilization, making stir culture, anaerobic fermentation, aerobic fermentation, and recovery. Same process. Okay. So another thing actually known as the glutamic acid. So glutamic acid is also very much important, and glutamic acid you can also produce the same process, same methodology. So that is widely actually used in both food industry and also in the general public as a flavor enhancing agent. I hope the girls are also aware, because you are actually watching lots of YouTube videos and other things. So you might have seen some people are actually using as a flavor this thing called your glutamic acid. Okay, so the high demand of glutamic acid actually increased rapidly before 1956. It was mainly maintained to the hydrolysis of the plant protein, but after that it has actually got popularity. It is also important in the brain metabolism, hence various analogs of the glutamic acid used to the treating various of the neuropathic diseases. That's why it is very much important. And also like PGA and other areas, it's also using as a polymer, as a non-toxic another substance. Okay, including monosodium, this thing called glutamate. It has been used as a flavoring enhancing agents by both food and this thing called general public. So MSG. I hope you can recall the MSG controversy with this thing called I think क्या नाम था उसका इसका जो मैगी यस मैगी में जो है the amount of monosodium glutamate it was actually so much high. That's why there are also controversy in 4 or 5 years back. Okay, so nowadays we're actually using glutamic acid. So how you can produce glutamic acid? Same process. Whatever I was actually showing here, same process, fermentation. Another thing actually, another substance is actually this thing called Griseofulvin. Okay, it can also used as antifungal antibiotic. It can isolate it from the penicillin species, and it is a secondary metabolite produced by the penicillin. Okay, so that substance also can be used as a various pharmaceutical purposes like ringworm beard, ringworm skull, and fungus diseases in nails, and ringworm area and athletic foot, including like ringworm body. So you can use this substance as a various antimicrobial drug properties. So what you can, how you can produce? Same process. Again and again, same process: fermentation, then pre-treatment of the fermentation, growth, filtration, extraction, decolorization. Yes, it might be newer for you. Decolorization you can also use. So many of the cases, many of the books you'll be observing they are not using decolorization, but whatever. So if you add, actually decolorization will require the color after extraction. Whatever the color you are getting, it is not so much good. That's why decolorization, and then isolation and separation, and lastly the precipitation and purification. That's it for today. Tomorrow I will tell you some basic things about the blood product.